Now we're recording. Sorry, I would hate to like skip the one job. We're going to start off just thinking a little bit about particle size distribution and why it might be useful. So we're going to start from the very bottom and build our way up, thinking about particle size distributions, and then work our way into optical proxies, which relate to volume scattering function and some of the methods you guys are going to start making this afternoon. But it's just sort of a useful base to have as we start thinking about some of the processes and measurements that we're making later on in the course. So what about a particle can we infer from its size? Or what might be some of the things that we are actually interested in? Say you're an ocean biogeochemist or biologist or something like that. Why do you care about particle size? Yeah. Biomass, okay. So how much living stuff is there contained in particles? Yeah.
Sinking speed or export.
Sinking speed or export, right? So if we start thinking about really big particles, they're going to have, you know, if they're not too draggy, right, they're going to have a sinking speed that's proportional to their diameter squared through Stokes' law, more or less. Yeah.
For the phytoplankton that you might have.
Okay. So it's one of those things that might help us distinguish between what kinds of phytoplankton there are.
Yeah, microplankton.
Okay. Microplankton, nanoplankton, picoplankton. I'm just repeating you because I don't think the camera can hear. But yeah, so phytoplankton types or functional groups might be related to their sizes. Yeah.
Rate?
Rate?
If it's alive. Like metabolic rate, respiration.
Okay, so metabolic rates, respiration, things like that might also be functions of organism size, right? For thinking about, especially zooplankton, animals, things like that. I don't know if that's what you're thinking about. But yeah. Yeah, Luca.
Potentially, first thing about the water, it's like nutrient levels.
Okay, so for single cells, the nutrient flux is going to depend on the surface area to volume ratio of the cell. So size is important for that as well. Yeah. Does anybody do sediment resuspension or particle transport work? Talk to anybody maybe who's done that. So if you're interested in sediment resuspension in the bottom boundary layer or sediment fluxes, those kinds of things also depend upon particle size, right? The degree that those particles are going to sink back down to the bottom and deposit will be a function of size. So there's a lot of reasons that we might care about particle size. I guess we kind of got into processes affected by size in that first question, so I'm just going to zoom through this and pose this one to you. What is the size of this thing? Duration, dinoflagellate. How would we determine that? So it's longest dimension, okay? So we could say, all right, from here to here is its size. What are some other ways that we might size this thing? Yeah.
By its general center. So
Okay.
just like a circle around the circle.
Okay. Yeah. So the size of this part. Yeah.
Into a sphere into diameter.
Okay. So oops. So the size of this, and then make it into a sphere, and then compute the equivalent spherical diameter of it. Okay. Yeah.
It depends what we mean by size. We're very biologically speaking, mass is important.
Right. So we could filter it and weigh it, or filter it and measure the concentration, the content of something in it. Right? Yeah, so there's a lot of different ways that we might actually try and quantify size, and we use a lot of different instruments to tell us size as well. We'll come back to this a little bit in a moment. You're not trying to raise your hand, are you? Okay. Sorry, people scratch in certain ways, and I think there's a hand creeping up. Okay, So what we're going to spend probably the next chunk of time on here is actually just stepping our way through how do we build a size distribution. There's a lot of questions to ask when you do this. So first off, how do you characterize a size, a particle size, and what aspects do these different observational methods that we have, what are they sensitive to? What units do we use? What are the uncertainties? How do we take what are sometimes discrete observations? And I'll start with a very simple example data set that you guys collected on Monday night. Right, so how do we get from a discrete set of particle counts to a continuous particle size distribution? And then how do we compare those if we have things from different instruments or different places, different size bins, things like that. And then thinking about number size distributions, volume, particle volume size distributions, area size distributions, going back and forth between those. It's not complicated math, but it's important to keep track of your units and make sure you're comparing apples to apples and thinking about what is your sensor telling you and what do you know and what do you actually don't know about the stuff that you're thinking about because of what your sensor tells you. All right, so my very simple data set that I'm going to just start us thinking about, and it's just because sometimes it's easier to have something concrete to think about as we start to move into this math, is the December 2020. I don't know which group of you guys did that one. I picked it because it seemed to have a lot of different things in it. It wasn't all just one or two classes. So I took some photographs and made a little mosaic. I went and I didn't count up everything in your smallest category. I just sort of measured what a block of 50 was, and then I estimated, there's about that many. And I counted up all the rest. And I started out, so you guys, how did you guys classify or make your frequency distribution of your particles for this? What's on your x-axis here? Species and organized by size, right? And organized by size across the whole year of data that all you guys had together, right? So if a particular taxonomic grouping was smaller in one month and bigger in another month, I assume you guys sort of figured out like, okay, it's roughly in this order. So in our December data set, things might not all have been exactly in order, but that's okay. That's what Excel's for. Okay, and I pulled out a couple of examples. I just did a little screenshot and made big versions of coccolithophores, and I think that says diddle just as examples, and we kind of talked through this already, but okay, size, right? The coccolithophores are kind of easy. I just went for diameter, more or less, and actually very, very rough, right? You'll see in a second. What about diddle? That's harder. It's got spines and it's very narrow in one dimension and wide in the other. I just ballparked the longest dimension. That is all I did because it was late last night and I couldn't really see the grain photograph very well. So it's okay. We just need numbers to start thinking about. Yeah, so this is what I did. If we were actually doing it the right way with the IFCB data, right? We would probably take those images and put them into some sort of an image analysis routine that would find the perimeter and maybe compute a volume and tell you, what is the enclosed area, what are the major and minor axes, something like that. And we'll come back to those ideas next week, I think, when we talk about image analysis. But so that's how we would do that with the IFCB. If we'd taken the same sample and run it through a Coulter counter, how many of you guys have heard of or used a Coulter counter? No? Okay, so Coulter counter is one of the older devices that's out there that's been used for sizing marine particles. And it's basically measuring the change in electrical conductivity when a particle passes through a measurement volume. And so it's sensitive to something that's kind of like particle volume. Okay, so that would tell you something different than looking at the projected area, which is what we're seeing from the IFCB, right? What would happen if we took that water sample and put it into a laser diffraction meter, like the list would learn how to use this afternoon? What would that be sensitive to? Controls how much light diffracts around the particle. What is it?
Size.
Size? What aspect of size?
Absolute.
Volume?
Orientation.
Yeah, so it might depend on the orientation. So diffraction is going to be sensitive to the surface area, some part of the area of the particle, right? So there's different aspects and actually the list, which is a laser diffraction instrument, might also give you some information depending on which way it's facing as well, or which part of the structure. So that serration that we saw on the first slide, right, might give you one response depending on the size of and I'm not a phytoplankton biologist, but little horn things versus the central sphere versus the entire enclosed volume. I'm sure it's not a horn, I know that, but somebody in this room actually probably knows what it's called. Or not. Okay, anyway, that's neither here nor there. Okay, so there's a lot of different ways we could get at size. Different instruments would tell us different things. I'm telling you here about the approximate length of the longest axis of the particle categories, eyeballing it. Okay, so super not exact. And what I made was just a little table. There's my camera. Of the categories and the number of particles that I either counted or for the smallest one estimated in that category, and then the size in microns. And then I sorted it by what I thought the size was. So they're not exactly in the same order that you guys put them on your x-axis of your plots, but that's okay because I knew you were sizing them based on the whole year's worth of data. Okay, oh, and I should note here that I'm using capital N in this case just to represent the raw counts, the raw number that I counted. Kurt just used that in the last lecture to talk about the cumulative number size distribution. So it's just using the same letter to me, for which I apologize. All right. So actually, I'll start here. What are if we sort of set aside the fact that I wasn't actually really accurately measuring particle size here, what are some sources of uncertainty that might go into these just counts for size? Sorry? So this is just size. So that's the same thing as me being bad at eyeballing the size from a composite photograph of your histogram, right? So let's assume that I'm perfect. You guys are perfect at cutting them out, and the sizes are correct. Yeah.
You're assuming that each organism is the same size within its class.
Okay.
Or grouping.
Yeah. So that kind of goes into the same bucket too of imperfect size measurements. But let's just set it aside for now. What about how big was our sample? How big was the water sample that actually generated these particle counts? 5 milliliters, right? So could there be some uncertainty associated with that? Yeah? Like, what if we took a different 5 milliliter sample an hour later? Or let's say 5 minutes later, actually, because the tide changes a lot here. Probably be similar, but not quite the same, right? Yeah.
Are you trying to get a rare species and whether you found them or not?
Right, or so we're talking about size really here, so rare sizes,
Rare sizes. Okay.
right? So like say there's occasionally a 500 micron cell that's floating around out there, but you would need to sample a lot to find it, right? So there are things that are undercounted. Yeah. Did you have?
I was going to say that if you have a small volume, then you're going to skew your distribution of probably the much smaller organisms.
Yeah.
Yeah.
Okay. Yeah. So if you have a small volume, you're gonna miss some stuff. We're basically collecting a random sample of a much bigger population, and so our counts should obey a Poisson distribution, right? And the uncertainty should be, if we assume that our measurement is the mean or the best estimate of the mean we're going to get, then our uncertainty is square root of n, right? So that means that if we were to just collect a bunch of 5 milliliter samples over time, we should find that our uncertainty in size in should follow that. Okay, so that's one thing to keep in mind, is that just the raw counting carries some uncertainty. There's also this issue that it kind of goes to what Ben was saying, that you can only count one particle or no particles. There aren't any, you need a bigger sample size to get to that particle that you're missing. There's some rare things where you don't get one, you really don't know, right? So there's this sort of raw detection threshold for rare things. There's also one other kind of uncertainty that I would put in here, which is if you look at that tiniest size fraction, the one that has over 1,000 particles in it, more or less, are they all 10 microns? Is it likely that there are some that might be smaller than 10 microns? Are there likely some that didn't get detected because they were so small, or it was not imaged? And actually, I don't know enough about the IFCD to know what that lower cutoff really is. But there's a lower detection limit to a lot of these particle sizing instruments as well. Okay, so there's a number of different uncertainties that are going to go in here. That counting uncertainty is what I'm going to propagate forward here. Okay, so we have our counting error, which should go with group n, where capital N is just the number of things counted. Our detection limit. At the small end, that's just our instrumental. And at the large end of our size spectrum, that's going to be determined by our sample size. We might just miss the rare things. Okay, and so before you even start to compute number of particles per liter, or number of particles per liter in a size limit, you should start propagating that counting error through. Right? You have to figure out that counting statistic error, that Poisson distribution comes from your raw counts. Yeah.
What is relative to that?
Because you can think of this as we're randomly subsampling something, a bigger population, and we're basically saying, okay, it could either be there or it can't be there, right? It's either yes or no, present, not present kind of sampling. And so sampling processes that are like that will have a Poisson distribution and the uncertainty there. Okay, so here's our size distribution again, our size counts, I should say. If we wanted to make a frequency histogram of the different particle size, the way particle size varies here, and we wanted to bin as a function of size, how would we go about choosing size bins?
I'm going to throw out the suggestion of every 10
Okay,
millimeters.
so take a regular increment. Okay, not irregular increments. Okay, that kind of makes sense. Yeah, so every 10, yeah,
If you do it by a certain percent,
certain percent.
like out of the like 1150 category 1 would be a certain percent of the total.
Okay, so you can normalize to your total amount. Yeah.
Whenever the logarithmic scale.
Okay, so maybe regular increment or use a log spacing. Why would you want to use a log spacing?
There's a huge range of values and it's a lot of
Yeah, so we've got like our smallest things, there's a whole bunch of them, and maybe there's fewer of them as we get to larger and larger sizes. It's going to make it easier to visualize and think about. There's some other reasons too that we'll get to. Okay. Oh, wow, I'm going really slow. All right. So we also want to make sure our size bins are encompassing our entire range of sizes, right? So in this case, I sort of took that approach, that log size spacing. So what I'm showing you here is just a screenshot of a real quick set of calculations. So over on the left-hand edge is exactly the same thing you've already seen, right? The sizes and the counts and everything. And then what I did is I made a set of size bins that are logarithmically spaced, space 10, and I found the edges of those bins first. And then I computed the center of the bin. Now, should I just take the mean of the two sides of the bin, their log space? No, right? So this is the bin center is the geometric mean of the two edges. So that would be the square root of edge 1, which I'll call D1, times edge 2, D2. Okay, so it's going to be a little bit smaller than the arithmetic mean, which would be the average of the two edges of the bin. Okay, so there's our size bin center. And then I've got the number, I just added up the number that fell between each of those edges. And then got my error associated with that. And I computed the relative error here, just to give you a sense for that 5 milliliter sample, for instance, for particles between 100 and 158 microns in diameter, which I kind of made up a little bit. The relative error is, verging on 50%, right? So that 5 milliliter sample doesn't get a whole lot of those. So if we're interested in particle size distribution, we've got some uncertainty out there at our large end with such a small sample size. Okay, so now we actually measured a volume of sample, right? So I've taken the numbers and I've computed just a really rough number distribution. So n as a function of d, diameter, and the units on that are going to be, and this is just to start, we're not going to stay here, but the number of particles per liter in each of these discretized size bins. And then we've got an error associated with each of those as well. But this is just discrete data, right? We just took a water sample, counted up everything in these different size bins, plotted it up. What if we want to compare this to a different measurement with different size bins? Or model using a continuous size distribution like the Young type or the power law distribution that Kurt was showing. How do we do that? Yeah.
Normalize to the bin size.
Right, so we need to normalize to the bin size to do that. So to get from our big ND, that's sort of our raw counts as a function of size. And actually, I'm going to do that per liter as well, since we're already in volume units here. We're going to divide by delta D, size of our bin. So that's going to be in microns. And we're going to get, I'm going to use the small n here to get our size distribution that's been normalized to bin size. And this is going to have units of, let's see, I've already written some wrong things, so let me fix it. This should be number per liter. There we go. So we're going to get to number per liter per micron. All right. And this is called a differential number size distribution. So it's independent of the bin width. We can compare across different texture measurements when we do it this way. And there is a typo right here. That should be little n. I'll fix it in the version that goes up online. Okay. Okay. So how do we go from a number size distribution, a differential number size distribution, to a volume size distribution? This might be important to think about if you want to, say, integrate, right? Figure out how much volume resides within each of these size bins. So this is just sort of asking you to think about units. Or maybe here's another way of thinking about it. The list, the laser in situ scattering in size.
These you say to scattering in size, transmissonimeter.
And transmissonimeter. Okay. The list is going to give us its output. If you're looking at size distributions anyway, you're going to get it in microliters per liter, or that's the same thing as parts per million. Those are the units it's going to give you size distribution in. So how do we get back and forth between this differential number size distribution and the power laws to error across steadiness instrument to instrument and this volume fraction, particles per water volume? That's what the list is. Our differential number size distribution is particle numbers per liter of water per micron bin width. How do we go from one particle to the volume of that particle? So we know we have one particle, we want to know what its volume is. We know some estimate of its size. We might want to make an assumption about what shape it is. Yeah, we could use a sphere. And we know we're not actually dealing with spheres, but we could start with that. So we could multiply by some conversion that's going to give us the volume of the particle per particle. This would be number of particles, just for completeness. Okay, so if that were a sphere, right, volume of the sphere is IV cubed over 6. You have a different shaped particle, you know that, you can do a different volume conversion. Okay, so we could get our particle volume, and let's just say, so that we'll compare to this output of the list, maybe we can figure out what the volume of the particle is in microliters. Maybe it's in cubic microns, maybe it's in cubic meters, whatever units you want to use. And then if we were going to get something that we wanted to integrate across, right, or compare to what the list is giving you, then we actually have to go back and multiply through again by our delta T limit, right? We will have the total amount volume of each particle type, each particle size class in each bit. Okay, so the list will be giving you something like that. That's how you would get back and forth. Okay? So it's important to just, like I said, it's not complicated math, it's just something where you want to keep track of your units and be thoughtful about how you're doing it. Okay, so let's talk a little bit about the actual ocean again. Stay away from the math, theoretical stuff. So this is a really old paper. This is from before I was born. It's one of the first to note this approximate power law distribution of the material in the ocean, this function of size. So Sheldon et al. In 1972 found that to first order, there's roughly equal amounts of material in log space size bins. They said from bacteria all the way to whales over 6 orders of magnitude. I don't know if that holds up across all ecological studies, and I think we're doing a lot more measurement of all of these now than we were at that point. But if you work that through, that's consistent with a differential number size distribution with a slope, power law slope of diameter to the -4. So that's where this sort of canonical Young-alike J-U-N-G this Young power law slope, that's where that comes from. This has sort of interesting ecological implications. Suggests that, if this is actually true, then maybe we should expect that growth rates should be inversely related to particle size, right? Since we have this transparent material up and down in size classes. So if you were to divide these bins by their bin width, you might get a flat volume versus size class distribution. So there's a lot of models that people use to represent particle size distribution. One of them we just have spent a bunch of time talking about, I think, in a few lectures. So this is the power law size distribution. I think in some older papers you might see it's called a hyperbolic size distribution, but it's not a hyperbolic function. So power law size distribution. So reference the number of particles at a reference diameter, which is d naught, right? We would take the diameter divided by d naught and raise that to this power law size exponent, which is usually a I think that's beta, right? Squiggle. And here we see this given again, numbers per volume per size bin. So this is a smaller size range than Sheldon was talking about with the tuna and the whales on it. But again, sort of you see if we plot diameter and the differential number size distribution on log scales, we get a straight line. We're not going to slope in that necessarily. There's a few more rare models that might actually be better in some situations. And in fact, there's no reason to expect that a power law distribution is actually the right one, sort of a priori in any setting, right? If you have a balloon going on, or if you have something that's producing a lot of particles in a particular size range, there's a process that's causing that to be enhanced. You wouldn't necessarily expect to see a power law. You might expect to see something like this, where you've got a peak, right? This gamma distribution that's superimposed maybe on a power law or present. So like the cultures that we measured yesterday, we get a size distribution of those. We might expect to see something like this. Right? So yeah.
Actually, the size distribution really holds a reference.
Yes.
If you pick one sample,
Yeah.
then you have probably not.
Yeah. Yeah, you wouldn't necessarily expect that. And in fact, if I go back to our size distribution, this is from December 2020 in the IMCB, sized by my eyeballs, so who knows, right? But you know, that clearly doesn't fit a power law perfectly, right? There's some variability in there too, which I think is probably real and not just me eyeballing things, right? So there's processes that are causing that. Like there to be less in the sort of 20-ish micron range and more in the 10 micron and larger ranges relative to a power law. So those little differences often contain interesting and important information. Here, okay. There's also models out there like this double exponential model. It's another less common one that sometimes gets used to fit the distribution a little bit better. This one has more parameters, right? There's two exponentials added together. And of course, any model that has more parameters is going to fit your data better. So you have to think about what the meaning of those parameters are when you start to fit real data. That's telling you. But this is another option here. So one really cool study that was back in the late 90s was George Jackson et al. Actually managed to make a size distribution that spanned 4 orders of magnitude in size by merging something like 5 instruments together. Right? So different instruments are going to have different ranges where they are sensitive to particles. There's different methods that they're all using as well. So in this particular study, the lower size range, the very smallest particles, were being counted with a Coulter counter, right? One of these impedance sensors measuring the change in electrical conductivity, so this is particle volume. And then as they got up into the larger size range, particles tens of millimeters in size, right? They were using imaging sensors, so cameras. So they here merged together the results from all of those. And on the left-hand side distribution, the left-hand side of the plot, we're looking at the particle differential number size distribution, right? Our units are numbers per centimeter raised to the -4th power, so that's a clue that they've normalized to bin size there. And there's a nice power law size distribution that comes out there. Looks like it fits pretty well, right? But of course, buried in all the little wiggles of that line around the power law are some interesting departures that actually are really important when, on the right-hand side of the panel, they convert to the volume size distribution of the particles. So here they have taken, in this case, they're actually waiting for some of the particles, especially the larger sizes or aggregates that have this fractal scaling, where they've got a lot of fluid included within the particle volume. So they are weighting and normalizing to the amount of solid stuff that's in the particles. So they've accounted for the packing of the material in these looser aggregates here. But you can see the weighted volume spectrum. So here this is the number of particles times their volume. So the volume per particle, that's where that weighting comes in. And then each bin has been multiplied by the bin width. So times D gives a measurement that's got ppm, parts per liter, microliters is particle volume per liter of water, etc. So you can see here, what's the difference between the, if you look just at the number size distribution, what sizes are the most important? If you look at the number size distribution. The little ones, right? But then when we look at the volume size distribution, particle volumes actually carry more in these larger particles. So there's some really interesting differences that pop out when you do that transformation. Depending on what you're actually asking questions about in the ocean, you may want to go one way or another. Even surface area distribution can sometimes be, or area distribution might be the thing that you are most interested in, depending on your application. So, important to think about there. You can also see, you know, this is a set of samples going down in the water column from 10 meters to 15 to 30. And on the number size distribution side, it kind of all looks the same, right? In fact, I think I would suspect that the spectral slopes, the power law size distribution slopes here are not significantly different from one another. They're all around 3. But then if you look over here and you integrate under the volume versus size curves, you can see there's in fact an attenuation of the amount of material, as you would expect, going down through the water. So there's some real important things that are buried in there. Okay, so sort of to take home, these different size methods are sensitive to different sizes, right? A Coulter counter is not going to be a whole lot of help if you've got centimeter-sized aggregates. But if you're interested in the solid size distribution of really tiny particles, it might be the way to go. And we're going to step forward here in a second to think about IOP proxies for size. We'll come back to the main theory, thinking about the sensitivity of attenuation, for instance, at particular wavelengths of particles of a particular size. So you can achieve these resonances with particles that have certain wavelength and size combinations. We've got these limitations of instrument resolution, right? So there's always going to be a small size below which we're not going to detect as well for all these instruments. You need to be aware of that, what kind of aliasing there might be. There's also this rare large particle issue, or sample size that's too small, right, to really resolve the frequency of the rarest particles. So those two limits, the instrumental resolution and sample size, are things that are worth keeping in mind. Okay, so we're going to start forward now, talking a little bit, and I've got 10 minutes left. We're stopping at noon to eat. Yes, we have to get back to Kurt after lunch. So 10 more minutes, that's perfect. So I'm just going to give you guys a few examples of some optical sensors for proxy, think of proxies for particle size. So one, this is almost to where we got to, yesterday at the beam attenuation lecture. But if you guys remember, we were talking about beam attenuation normalized to particle volume and how there's a peak in the sensitivity of that IO peak as a function of particle size. It turns out that you go through mean theory calculations and actually see this in the environment as well, which I'll show you in a second. That peak size sensitivity varies as a function of wavelength. So at blue wavelengths, that peak sensitivity is at smaller sizes, and it's a little more constrained spectrally. Sorry, the size range is more constrained, and then as you go towards larger wavelengths, the peak sensitivity is for larger particles, and there's a broader sensitivity there. So spectral beam attenuation. Yesterday we really only talked about single wavelength beam attenuation. Start to get spectral BMC, there's going to be size information in the spectral shape of beam attenuation. So this comes out of me modeling, and it works pretty well for particles sort of in smaller size range. So the top equation there is just, again, this power law particle size distribution we were just talking about. The number of particles is governed, the number of particles as a function of size is governed by this power law function, and our size exponent eta. So mean theory tells us that we should see a relationship between the power law size exponent and we fit same power law function to our beam attenuation spectrum. The slope of that should be related through this fairly simple relationship. Eta equals gamma, where gamma is the beam attenuation size distribution slope, or sorry, the beam attenuation spectral slope, plus 3. And there's actually a small correction that can go in as well if you're at the very low end of the range of that. But for the most part, just the eta equals gamma plus 3 is the expected relationship that comes out of mean theory. And we can see qualitatively, here's some data from a series of deployments that Emmanuel and some of his colleagues did now about 20 years ago. But basically they measured the spectral shape of particulate beam attenuation in the top panel here. And in the bottom panel, these are data from the Coulter counter. So again, this is the electrical impedance measurement of particles. So the data density is much smaller because the Coulter counter analyses are just bottle samples, discrete, done by hand. But both of them are plotted versus the particulate beam attenuation magnitude at 660 nanometers. And you can see that there's this, they both have the same power law slope versus attenuation dependence. And in fact, if you look just sort of roughly on the y-axis, there's this +3 relationship between them. They even both show this little cluster of particles that were collected in the first stage of sampling after irradiation, right? So together we see those size and beam attenuation spectral slope properties change. So we can see this also in a bottom boundary layer measurement. So if you're near the seafloor, you've got sediments down there. If you have an event that resuspends a lot of sediment, right, you can pick that up off the bottom. But the particles that are larger, this relationship between settling speed and the square of the diameter, those larger particles will settle out faster. So we would expect to see, in general, a relationship between the total attenuation and the particle size distribution as you go down towards the bottom, in the bottom of a turbid crystal area. And in fact, this is what they saw. This is the same, I think the same study as before. And basically what they're looking at is this bottom boundary layer optical property distributions at 3 different dates. So this middle one here is right after a hurricane passes. So there's this event that picks up stuff off the bottom. And you can see that there's this increase in the beam attenuation coefficient in the middle panel. If you just look at the x-axis, you can see that the scale is totally different in the middle one, right? So increase in the attenuation by all these resuspended particles. And this value of gamma, so higher values of gamma are going to tell you that there's smaller particles, lower values, generally larger particles, because that's what's going with the particle size distribution spectral slope. So again, you can see those values shift right after that hurricane comes through the middle panel, all the way on the right here has much flatter values of gamma that are consistent with this resuspension of large particles on the bottom. And then come back a different day when that isn't the case. Actually, this is back in time. It looks like, but regardless, like you've got these settings that are not hurricane passage settings, where both the beam attenuation is lower and the particle size distribution slopes are similar. And you can see that change if you go up off the bottom as well. So it's flatter, closer to the bottom, steeper, smaller particles as you come up away from the bottom. So I think Colin showed you this yesterday or something like this, right? So this is just sort of a review that as particles get bigger and bigger and bigger, you've got more of this contribution of diffraction around the particle and this near forward intensification of the volume scattering function. And so we see that beta gets very peaked in near forward scattering angles. So this also can be inverted to give a particle size distribution. I think today you're just going to be using the actual VSF data and thinking about how the shape of the volume scattering function changes, and just doing sort of the scattering measurements. But eventually you can use this as particle size proxy as well. So in the bottom boundary layer, we see this closure between the list particle size distribution measurements and the gamma spectral slope of beam attenuation, and also actually the spectral slope of backscattering. Now, why might we expect or not expect backscattering spectral slope about particle size? What else might affect the shape of the backscattering spectrum? Wavelength. Absorption, right? So if you have particles that have, like phytoplankton, that have absorption bands, remember scattering will decrease in the wavelength bands where you see absorption, right? And we don't really know, like backscattering is going to be similar, right? But if the backscattering spectral slope also has these absorption bands, then we wouldn't necessarily expect that same correspondence. But these measurements were collected in the bottom boundary layer where it's mostly suspended sediments and there aren't strong absorption bands. And so we do see this good agreement between the backscattering spectral slope and the average, average particle diameter as measured by the list with the near forward VSF conversion. Eta is from the list. Yeah, and that's not even on here. They're just plotting versus this inverted particle size. Yeah. So it's still kind of an open question whether the backscattering spectral slope is going to tell us as clearly what the subparticle size distribution is because of this influence of absorption. It's used in particle functional type inversion models for remote sensing, so it's a very important question, but we're still working on validating. Another IOP-based proxy for particle size is this method that Nathan Briggs came up with, also about 10 years ago. So the first kind of thing that he observed is that if you take a profile of single wavelength backscattering or fluorescence actually, or beam attenuation, pretty much any IOP. And it's measured at really high resolution and unfiltered, it's full of spikes. And people had seen these spikes in for a while and thought, oh, this instrument was filtered enough. Well, what he found is that if you filter and actually separate the spikes from the baseline signal, right? So you've got sort of your background backscattering signal, and then the spikes in the backscattering. Those spikes actually seem to be related to large particles in the water column. So what he found was that you collect a series of backscattering profiles in a Lagrangian mode, so following a water parcel. In this case, it was during the North Atlantic bloom experiment in 2008. So they were following a patch of water through the spring bloom. You could see that the backscattering spike signal, which is the color field in the contour plot here, showed this, especially below about 200 meters, showed this plume of large particles going down into the mesopelagic over a period of maybe 10 days at the end of the bloom. So this was sort of the first application of this, and there have been a few papers prior to that that have suggested that spikes in optical properties could control large particles or zooplankton, things like that. But this was one of the first sort of saying, look, you can actually use this as part of this. It's not noise, it's an unexplained thing. It turns out it's an opportunity, it's a way of telling us something about processes in the ocean. He went on to actually come up with a calibrated size proxy for particles. So if you can imagine you've got backscattering sensor, shoots some light into the water, and then it comes back to a detector through a defined angle, right? You have a little particle that passes through that. It might make a little spike. It might occupy that beam for less time. If you have a big particle, it's going to occlude the beam more and for longer. And so what he found is if you take the variance, so how much noise there is, right? Noise, not actually noise, signal. The variance of your backscattering signal versus time, and compare that to the mean, and adjust that for the residence time of water in the sensor's optical sampling volume to the sample integration time of the sensor, you can get an estimate of particle size, the mean particle size of whatever the sensor is looking at. So the figures here are showing the comparison of this variance to mean ratio method of particle sizing with the list gives from its near forward diffraction, laser diffraction measurements. So you can see that over a given size, there's sort of an optimal size range for this, right? But it works for backscattering, it works for beam attenuation and AC9, and it works for beam attenuation in the list, right? So he was able to show that this works for a lot of different optical sensors. It's a little bit difficult to apply in the field because you need to be moving your sensor very slowly relative to its integration time, right? If you put a C-star transmissometer on a ship's rosette and you profile it at 60 meters a minute, you may, or the ship's rolling up and down, it's going to be harder to interpret that. But in a nice slow profile or in a controlled laboratory setting, with like gliders or floats, things like that, would be a good method. And Nathan's actually gotten some neat additional information from this proxy recently, getting the DBP in the lower panel is the mean particle size. This was on a glider following that same North Atlantic bloom parcel of water around. And he sees this peak and then drop in the mean particle size distribution over time that coincides with this peak in the bloom as measured by other proxies for gross and net primary production and export, and then this crash that happens after silicon depletion happens. So some sort of a neat application of that more recently. So in summary, size clearly matters. We all know this for a lot of different oceanographic processes, but you have to define it. It's important to keep track of your error, keep track of your units and be cognizant of the sensitivities of the instruments you're using, and, you know, whether your sample size is limiting your rare particle collection, or whether you're aliasing or missing some of your smaller particles of interest. As Kurt just said, all data looks good on a log plot, including size. So yeah, details are important, and there's a bunch of different optical proxies out there that are useful for constraining particle size. So all I've got is lunchtime. That's it. We've got some scattering models.
Excellent.